Rabbits were first domesticated around 1,400 years ago in southern France. Since then, they spread worldwide, and today more than 400 rabbit breeds are recognized globally. These animals are not only important for producing meat and fur, but also serve as a valuable model in biomedical research. China, in particular, plays a major role in rabbit breeding, in that the Chinese rabbits are known for their strong reproductive performance, disease resistance, and ability to survive on rough feed. However, due to small population size and intensive directional selection, where breeders repeatedly select for specific traits, these populations are now facing a high level of inbreeding. Inbreeding increased homozygosity means less genetic variety. When that happens, usually useful traits get lost and the populations become weaker over time. To study this issue, we look at something called ROH, runs of homozygosity. Think of RH as long as stretches of identical DNA inherited from both parents. They appear when their shared ancestry, either recently or many generations ago. Short ROH often mean distant inbreeding from way back. Long RH suggests more recent inbreeding, like close relative mating. RH help detect hidden harmful genes linked to weak traits, which is why this study used FROH to help more accurate measuring of inbreeding. A genomic measurement for doesn't rely on pedigree. RH have been studied in animals like cattle and pigs, but not much in Chinese rabbit breed. In the study, research sequenced 170 rabbits from 11 breeds, 7 local and 4 exotic. Using tools like PearLink, JTK, and BWA, they identified over 31,000 ROH from 15 million genetic markers. They also look at ROH Island, regions where ROH appears often across rabbits. These areas may show positive selections, where breeders unknowingly fix certain traits. The meta plots show these ROH hotspots. Overall, the study helped us understand how inbreeding and selection shape rabbits' genetics and supports better breeding and conservation in the future.
For the first method, sample collection, sequencing, and SNP calling, a total of 117 rabbits was used from 7 Chinese breeds, 3 European breeds, and 1 Japanese breed. Blood samples were collected from the marginal auricular vein, and the DNA was extracted using phenylchloroform method. DNA quality was checked using nanodrop spectrophotometry and agarose gel electrophoresis. DNA samples were used to construct 2 paired N libraries per sample, and sequencing was performed on the Illumina NovaSeq 6000. Raw sequencing data was filtered using FastP for quality and aligned using BWA tool, and duplicate was removed using Picard. Variants were identified using haplotype column module and GVCF with GATK. ROH, or runs of homozygosity, is identified using Blink. The parameters used are shown on screen. Genome inbreeding coefficient obtained as a proportion of the autosomal genome covered by ROH using the following equation. Additionally, the genomic inbreeding coefficient based on the observed and the expected number of the homozygous genotypes was calculated using Blink using the second formula on screen. The GON software was employed to estimate the recent effective population size for each breed. All simulations were run for 2000 generation calculated in 400 bits. Each simulation was repeated 40 times with the default settings to account for variance. Heterozygosity reflects the probability of a locus being heterozygous and serves as an indicator of population genetic diversity. Both observed heterozygosity and expected heterozygosity was calculated using Blink. ROH islands is defined as genomic regions with a high frequency of overlapping runs of homozygosity across individuals. Blink and BAIT tools is used to detect and delineate these consensus homozygous regions. Enrichment analysis is performed through DEWIT using both KEGG and GEO databases. Functional categories with FDR adjusted p-values less than 0.05 were considered statistically significant.
Now, I will explain one of the key findings from the study, which focus on the distribution of runs of homozygosity, or ROH, in different rabbit breeds. So, after filtering the genome data, the researchers identified approximately 15.37 million SNP. So, these are genetic variation across the autosome, which are the non-sex chromosome of 170 rabbits from 11 different breeds. So, using this SNP data, they detected a total of 31,429 ROH segments in all the rabbits combined. So this bar charts here shows the number of ROH per breed, and it clearly highlights some important difference. For example, the Deutsche rabbit, which is a Chinese indigenous breed, had the highest number of ROH, with around 7,476 detected. On the other hand, the Flemish giant rabbit, which is an exotic breed, had the lowest ROH count, only about 1,316. This big difference actually tells us something very important. A higher number of RH usually means more inbreeding or stronger artificial selection within that breed. Continuing with the distribution of RH, this part of the study looks at where in the genome these RH are found and how long they are. So, researchers used a genetic tool called PLINK to identify and categorize RH by their length. They found that RH were most frequently located on chromosome OCU1, OCU3, OCU2, OCU7, OCU12, and OCU14. Among the rabbit breeds, we can see Deutsche and Japanese white rabbits had the highest RH presence, while the Flemish giant rabbit had the least. This graph here shows the number of RH across all chromosomes split into 4 line categories. You can see that short RH, which lie between 1 to 2 MB, dominate across almost all chromosomes, especially on OCU7, which had the highest number of rows. In contrast, long RH over 6 MB are very rare, making up only about 0.83% of all RH. This result suggests that most of the inbreeding happened in the distant past, since long RH, which reflect recent inbreeding, are rare. Now, I will explain the inbreeding levels and RH islands found in the study. First, we look at FRH, which is the percentage of the genome covered by RH. A higher FRH means more inbreeding. Across all rabbits in the study, the average FRH was 0.23. The highest FRH values were seen in Japanese white rabbit with 0.38 and Deutsche rabbit with 0.28, while the lowest FRH values were in Flemish giant at 0.14 and pigeon yellow and Laibu black both around 0.16. So we can see that some breeds have undergone much more inbreeding than others, depending on their selection, history, and inbreeding practice. Now moving on to the ROH islands and their gene functions. ROH island is a region where many individuals have overlapping RH segments. These are usually signs of positive selection. In other words, important traits were being selected in that part of the genome. So in Chinese indigenous rabbits, 17 ROH islands were identified, ranging in size from 0.25 to 3.53 MB. These islands were located on 7 different chromosomes. The largest RH island was found on OCU9, named RH9. It was 3.53 MB long. These RH islands likely contain genes that were selected for reproduction, survival, or adaptation, which are very, very important traits in native rabbit breeds.
So hi, my name is Swan Isora, and today we would like to discuss about the further investigation on this article, which we can see from the Manhattan plots that shows the percentage of individuals which each SNP falls inside an ROH. The x-axis represents the chromosome, while the y-axis represents the percentage of individuals with that SNP in a ROH. From this plot, we can see the ROH region for Chinese indigenous rabbits, where 70 ROH islands were identified that contains 1 to 4 genes, and also there are several high-frequency ROH islands that can clearly visible on chromosome OCU17, OCU12, and also OCU14. As we can see, there's also the longest island, which is on OCU9, which is 3.53. Examples of gene is reproduction genes such as motility or saturations, and also adaptation genes such as olfactory signs and also temperature tolerance. Furthermore, as you can see from figure 3b, shows the ROH region for the exotic rabbits, where we can find 22 ROH islands were detected, and they contain 186 genes, where the longest island falls on OCU7, which is 4.23 MB. This region contains genes for growth and fate traits, which common targets in commercial breeding. Example of gene is such as fat deposition, body growth, weight, and also metabolism. Most ROH that found were short, which is 1 to 2 MB and spread across the genome. So it shows that the Japanese white and Asian rabbit had the highest number and length of ROH, which is most likely due to the strong recent selection or bottlenecks. Other than that, Flemish giant and also Fujian yellow rabbit had the fewest ROH, that suggests that they are more genetically diverse or have larger breeding population. For the conclusion, ROH are common and found all over rabbit genome, but most of them are short and the long ones are not seen very often. So this shows that rabbit are most likely experienced inbreeding from a long time ago. In conclusion, they have detected a few things such as 17 ROH island is found in Chinese indigenous rabbit, while 22 ROH island is found in exotic rabbit breeds. Genes related to reproduction and adaptation is found in Chinese indigenous rabbit breeds, while the genes related to fat deposition, growth, and weight is found in exotic rabbit breeds. So lastly, we would like to talk about the purpose of studying ROH, which is can help to improve the rabbit breeding by optimizing the selection schemes, reduce the negative effect of inbreeding by reduce the inbreeding depression, and also support sustainable utilization of rabbit genetic resources by making sure that rabbit breeds are used wisely and healthy for the future. I think that's all from us. Thank you.